Welcome to this deep dive. Today, we're taking a truly fascinating journey. We are starting from the fundamental basics of molecular biology and going all the way to the absolute mind-bending frontiers of paleovirology. Whether you are completely new to the microscopic world or you're a seasoned virology researcher keeping up with the latest studies, prepare to have your perspective entirely transformed. It's a pretty provocative question, isn't it? I mean, we typically view viruses purely as pathogens, invaders that we absolutely need to eradicate. But what if the very things that make us sick are also responsible for making us human? Today, we are unpacking a massive paradigm shift that's going to challenge everything you think you know about viruses. We're pulling directly from cutting-edge research to show how these tiny, mysterious entities have literally shaped life on Earth. Okay, let's dive into this. Part 1. The borderline of life. We've all heard that classic high school biology debate, right? Are viruses even alive? Well, to really understand this, let's contrast their traits. On one hand, viruses possess two key characteristics of life. They contain genetic material, either DNA or RNA, and they can reproduce and evolve. But look at the other side of this comparison. They have 6 distinctly non-living traits. They're acellular, meaning they lack cellular organization. They have no metabolic machinery of their own, no autonomous reproduction, they don't grow in size, they don't respond to environmental stimuli, and, remarkably, they can actually be crystallized into solid structures, just like salt or sugar. So if you've ever been confused about this, your confusion is totally valid. To biologists, viruses act as a very weird borderline between simple chemistry and complex life. So to bridge this gap, modern researchers often use what's called the virocell model. It's a framework first proposed by Patrick Forterre. This model suggests that the independent virus particle you picture in your head, the virion, is actually just a dormant spore. The true living form of a virus is the infected host cell itself, actively producing new viral components. It totally flips the script, but that begs the question, how exactly do these dormant chemical spores wake up and take over a complex living cell? Which brings us to part 2, the cellular hijack. Let's break down the highly coordinated molecular virology behind this cellular takeover. You can really think of viruses as the ultimate cellular hijackers. First is attachment, where viral proteins bind to specific receptors on a host cell. This determines the virus's tropism, or basically which specific cells it can actually infect. Then comes penetration, often through a process like endocytosis, followed by uncoating to release the viral genome. The fourth step, replication, is where the real magic happens, followed by the assembly of new virions, and finally, release. For the virology researchers tuning in, you already know the immense biochemical complexity hidden inside these 6 steps. But for everyone else, the absolutely crucial takeaway here is just the sheer mechanical precision of this entire process. Now, what's really interesting about this is the realization that viruses don't just attack a cell, they rewrite it. They rely entirely on the host's polymerases and ribosomes to do all their dirty work. They are master manipulators overwriting the host's genetic program. And as we're about to see, this highly intimate genetic hijacking doesn't always just end with a destroyed cell. It can have permanent, long-lasting consequences for the host. All right, let's pivot to part 3, the evolutionary engine. To really see viruses as the architects of biological diversity, we have to look at a primary mechanism called horizontal gene transfer, or HGT. Now, unlike vertical gene transfer, which is simply you inheriting traits from your parents, HGT is the transfer of genetic material between completely unrelated species. You can sort of visualize viruses as nature's genetic flash drives. They are constantly plugging into different organisms, copying files, and then pasting those files into totally new, unrelated hosts. In fact, as our sources highlight, viruses are literally one of the largest reservoirs of unexplored genetic diversity on Earth. Take bacteriophages, for instance. Those are the viruses that infect bacteria. They do this constantly. They can actually integrate highly beneficial DNA, like the genes for antibiotic resistance, directly into a bacterial genome. This doesn't just change the bacteria on an individual level. It rapidly accelerates evolution on a massive, population-wide scale. And all of this creates an intense biological arms race. Viral infections are constantly introducing massive new genetic variations into populations. Surviving these constant viral onslaughts forces the host organisms to adapt, which is exactly what drove the creation of our own highly complex immune systems over millions of years. But it leaves us with a really fascinating question. What happens when that viral DNA doesn't just challenge the host temporarily, but becomes a permanent, working part of it? And that leads us to the most mind-blowing twist of this entire exploration. Part 4, how viruses made mammals.
Get ready, because this really changes how we view our genetic origins. We uncover this history through a relatively new field called paleovirology. It's exactly what it sounds like, the study of ancient, extinct viruses. But researchers aren't digging in the dirt for these fossils. They are looking deep inside our DNA. Millions of years ago, certain ancient retroviruses actually embedded themselves into the germline cells of our mammalian ancestors. And incredibly, those viral sequences were passed down generation after generation, right to us. And this brilliantly illustrates the result of one of those ancient integrations. Notice the syncytial cell layer right here at the fetal-maternal interface, what we know as the placenta. Researchers discovered something incredible. A captured viral gene, which we call syncytin, is actually responsible for allowing these cells to fuse together to form this vital barrier. This, my friends, is a textbook example of exaptation. It's when viral genes that originally evolved simply to help a virus fuse with a host cell and multiply were somehow captured, domesticated, and then totally converted into genuine cellular genes with an essential physiological role for the host. The staggering, almost unbelievable reality here is that without this domesticated ancient retrovirus, live mammalian birth, and therefore humanity itself, literally would not exist.
Let's make this abstract genetic concept a bit more tangible. Look at the physical anatomy of this mouse placenta, specifically showcasing the syncytio-trophoblast layer. It visually demonstrates exactly how those ancient viral envelope proteins were co-opted. They were repurposed to create the highly intricate biological architecture necessary to sustain fetal life inside a mother. But here is the real kicker. This wasn't just a one-time lucky genetic fluke. What we're looking at is convergent evolution in action. Researchers have found that totally different mammals, humans, mice, sheep, and rabbits, all acquired different retroviral envelope genes completely independently of one another at entirely different points in evolutionary history. It's like nature repeatedly found that capturing viral DNA was simply the best possible way to build a placenta, which leads us perfectly into our final and highly debated theory.
Part 5. The Cosmic Origins Theory.
So where did some of this ancient world-changing genetic material actually come from in the first place? This outlines the deductive and inductive methods that researchers use to formulate what's known as the cometary panspermia hypothesis. It is an objective, rigorous scientific framework that predicts that the sudden evolutionary jumps we see on Earth might actually be driven by the introduction of novel genetic material arriving from space. The mechanics of this cometary panspermia hypothesis suggest that extraterrestrial viruses delivered to Earth via comet impacts over billions of years managed to survive and become incorporated into the germlines of earthly organisms. It theoretically proposes a sort of galactic-scale horizontal gene transfer, constantly seeding planets with new evolutionary potential. Now, while the cosmic space delivery method is definitely debated in the scientific community, we can absolutely connect the core logic of this theory back to undeniable, observable biological facts on Earth. Survivors of major pandemics do carry DNA of retroviral origin. Our genomes bear the unmistakable testimony of a history of prior viral infections. In fact, human DNA is absolutely packed with viral sequences acquired over billions of years of evolution. So as we wrap up, I want to leave you with this really powerful thought to ponder. If the literal code of life is constantly being rewritten, shuffled, and upgraded by viral invaders, are we ultimately just a complex mosaic of ancient viruses? It completely changes how we view ourselves, our history, and the microscopic world around us, doesn't it? Thank you so much for joining me on this deep dive. And remember to keep questioning absolutely everything you learn.